A cat, seemingly innocent source of comfort, carries a hidden passenger. A simple meal, prepared without precise care, becomes a conduit. These seemingly disparate elements converge in a symphony of biological disruption, a silent orchestration of a change within the human host. We are exploring the shadow realm where Toxoplasma gondii and foodborne pathogens collide, where the gut-brain axis compromised by dysbiosis acts as a volatile catalyst and where boundaries between physical and mental health blur. This is not a tale of science fiction, but a compelling exploration of the biological forces shaping our lives, our mind, and our very essence. We are not just discussing theoretical connections, we are confronting a reality where microscopic invaders may be influencing our decisions, our mood, and ultimately our very personalities. So join us as we reveal the surprising truth about this parasite, a brainwashing parasite that infects nearly one-third of the global population, manipulating our behaviors.
Hello everyone and greetings from Egypt. I'm Hanem Khater, a professor and head of the Department of Parasitology, Faculty of Veterinary Medicine, Banha University, Egypt. Thanks to the organizing committee for inviting me to talk about a very interesting and shocking subject as well. Converging pathways, latent toxoplasmosis, and pediatric neurobehavioral outcomes. This talk is exclusive for the 4th World Conference on Pediatric Neonatology and Infectious Diseases.
Puppet master known as Toxoplasma gondii has long been a subject of fascination for scientists and cat lovers alike. It's one of the major causes of infectious reproductive failure and the second leading cause of death from foodborne diseases in the United States. It leads to behavioral changes that affect both humans and animals. So stay with me as this day is full of surprises. The prevalence of toxoplasmosis in humans is staggering. Estimates suggest that up to 30 to 50% of the world's population might be infected, with rates varying widely between countries and cultures. This means that billions of people could potentially be influenced by parasites they don't even know they have. To understand this teeny troublemaker that might pulling our strings, we need to take a closer look at Toxoplasma gondii itself. This unicellular parasite got its fascinating life cycle that reads like a bizarre science fiction, as we see. The very host of Toxoplasma gondii are cats, both domestic and wild. These feline friends are the only animals in which parasite can sexually reproduce, making them crucial to its survival and spread. But here is where things getting interesting. Toxoplasma gondii has evolved to infect a wide range of warm-blooded animals, including humans as intermediate hosts. It's worth to mention that oocysts disseminated in cat feces are highly infectious and extremely stable in the environment. Well, how does this crafty creature makes its way to our body? In fact, there are several routes of transmission, each are more unappetizing than the last. The most common way by which humans get infected is by ingestion of undercooked meat containing Toxoplasma gondii cysts, contacting cat feces which contain infectious oocysts. Gardeners take note, soil contaminated with these oocysts can also be a problem, as can unwashed fruit and vegetables. Risk factors for contracting toxoplasmosis include owning a cat, especially if you are not fastidious about litterbox hygiene, enjoying your steaks on the rare side, and having a compromised immune system. Pregnant women are also at high risk as the parasite can cross the placenta and potentially harm the developing fetus. Toxoplasmosis forms. Acute toxoplasmosis is a relatively mild and short-term disease. Develops immediately after infection, which is usually accompanied by swollen lymph nodes in the neck and a range of more or less severe symptoms resembling those of tonsillitis. In most immunocompetent individuals, acute toxoplasmosis spontaneously goes to the latent phase. Latent toxoplasmosis is characterized by lifelong presence of the dormant stage of the parasite, the bradyzoite, during which clinical symptoms disappear, but the parasite persists in tissue cysts in various organs of the intermediate host and prefer the neural and muscular tissues. Cysts persist likely for the host's entire life. The vast majority of infected individuals never find out they have had toxoplasmosis, and a small number of individuals with immune disorders or other predispositions, toxoplasmosis can progress to the chronic form characterized by some clinical symptoms of acute toxoplasmosis that persist for a long time and reoccur repeatedly. Naturally, rats have an innate fear of cat urine. Therefore, they run away from places that has cat. But infected rodents is attracted to cat urine, as you see in this maze. Toxoplasma gondii can cause permanent behavioral changes in its host even as a consequence of adult-acquired latent infection. Such behavioral alteration appear to be the product of a strong selective pressure for the parasite to enhance transmission from its intermediate host reservoir, primarily rodents, to its feline definitive host, wherein sexual reproduction can occur and the life cycle completed. Initial studies observed that laboratory mice inoculated with Toxoplasma gondii show significantly diminished learning capacity and memory in double training-based experiments compared with their uninfected counterparts. This leads to high predation rate, a more specific method for Toxoplasma gondii to increase the intermediate host activity because cats can immediately attract it to moving objects and exposed objects and show little interest or can see stationary ones. A series of interesting studies investigated the potential effect of postnatal and congenital toxoplasmosis on laboratory mouse activity and explored behavior by recording each individual's entry into marked squares on a cage floor, Y-shaped maze, and/or running wheels. Infected mice were found to be more active than their uninfected counterparts. Likewise, infected mice showed a preference for more exposed or novel areas of the apparatus and spent significantly less time grooming, a typical displacement activity before investigating such novel areas than did their uninfected counterparts. Rats showed increased activity, impaired learning ability, easily captured by traps, in addition to prolonged reaction times. It's clear that a slowed down mouse would find its way into a cat's stomach much more easily than normally agile mouse. This means that Toxoplasma gondii manipulates its intermediate host into its favor to complete its life cycle.
Okay, what about behavioral changes in animals other than rodents? Serologically positive wolves showed dispersal and assuming the role of a peak leader. Infected chimpanzee showed a loss of innate aversion toward the urine of their natural predators. They more frequently approached and investigated leopard urine than non-infective individuals.
In 26, I was doing a research on toxoplasmosis in goat, and I read in a paper that toxoplasmosis is linked to schizophrenia. What? I was shocked and said maybe it's a mistake, but I dig deeply about such relation. I found behavioral changes in infected people as well. In fact, whenever I read about toxoplasmosis, I always found new and fascinating information. That's why I am with you today to present the latest update about toxoplasmosis and its relation to behavioral changes and gut microbiota. In fact, latent toxoplasmosis lead to changed personality traits. Infected individuals may exhibit increased levels of impulsivity, aggression, and neuroticism. Neuroticism is a core personality trait characterized by personality instability, irritability, anxiety, self-doubt, depression, and other negative feelings. On the flip side, they might also show decreased levels of consciousness. It's important to note that these changes are typically subtle and may not be noticeable in everyday life. Anxiety. Everybody suffers from anxiety. Markovic and his colleague found that toxoplasma seropositivity was associated with generalized anxiety disorder, GAD. A 2-time greater odds individual in the highest antibody level category had more than 3 times higher odds of GAD. Watch carefully. Prolonged reaction times. The analogy of the predation accident. Approximately a 2.7 times higher probability of an accident in infected individuals compared to uninfected individuals. The increased probability of traffic accident applied to both infected drivers and infected pedestrians who were hit by a car. Among infected individuals, the lowest risk of traffic accident was found in those with the lowest antibody titers, likely indicating a long-ago infection. The risk was higher in those with medium rates and the highest in those with the highest titer, suggesting a more recent infection. Additionally, it has been shown over time that toxoplasmosis also increases the risk of workplace accidents. Individuals with toxoplasmosis are more likely to engage in risky activities from extreme sports to reckless driving. It is as if the parasite is whispering, "Go on, take the chance," in the back of our minds. Since the correlation is the opposite for the reaction times, with the reaction times gradually worsen with the time since infection, it's likely that infected individuals gradually adjust their driving styles to compensate for their impaired reaction times due to toxoplasmosis. The effect of latent toxoplasmosis on physical resilience and endurance. Infected individuals tend to give up prematurely. Again, this could be a trait that helps an infected mouse end up in a cat's stomach. In 3 independent studies, male Toxoplasma gondii infected students and faculty reported that when they are physically attacked and started to defend themselves, at a certain point, they experience a sudden impulse to stop defending themselves, as if their subconscious mind urged them to surrender. Toxoplasmosis is particularly associated with altered decision-making, which could lead to risk-taking behavior, thereby increasing the likelihood for suicide behavior. Similar to infected mice, humans might also be more likely to show risky behaviors when carrying latent toxoplasmosis. For instance, not wearing a helmet while riding a motorcycle, engaging in traffic accidents, drug abuse, or attempting suicides. Toxoplasmosis play a role in suicide behavior by altering decision-making process toward riskier choices. Of course, there are behavioral differences between sexes. In women, some factors decrease, like testosterone, impulsivity, subconsciousness, and pretension. These factors increase in men. Actually, increased testosterone, impulsivity, subconsciousness. Pretension means think of themselves as more impressive, successful, more important than they really are. Therefore, such infected man is not preferred by women. More sex-specific opposite behavioral and physical reactions are shown in case of infection. All these factors increase in females and decrease in males, like the following: clothing tidiness, super ego strength, aggression, self-control, affectothemia, means warm, outgoing, attentive to others, kindly, easygoing, participating, like people. In experimental games, generosity and altruism. So females spread kindness like Nutella. What happens next? Schizophrenia. Fact or fiction? Arias et al. In 2011 made a meta-analysis of 38 studies of the Toxoplasma cerebrans in schizophrenia and found that the likelihood of infection in schizophrenia patients to be 2.7 greater than that of the general population. Also, Freger in 2025 indicated that individuals who had contact with cats in childhood are at the increased risk of schizophrenia, and that many antipsychotic drugs used to treat schizophrenia inhibit the reproduction of Toxoplasma gondii. Schizophrenia in infected and uninfected individuals may represent two quite different diseases. First of all, the so-called positive symptoms of the disease, delusions and hallucinations, are more frequent and severe in infected individuals than uninfected individuals, and the hospitalization of schizophrenic patients with toxoplasmosis is also significantly longer in average. The most interesting difference, however, is that the changes in the internal structure of the brain characteristic of patients with schizophrenia, namely reduce the gray matter density in certain areas of the brain, occur only in patients infected with toxoplasma, not in uninfected ones. In this figure presented by Fleger in 2025, the difference in green matter density between toxoplasma infected and uninfected individuals with schizophrenia on the top half and without schizophrenia in the bottom half. Colored regions indicated areas where gray matter density is significantly lower in infected individuals compared to uninfected ones. Data were obtained using magnetic resonance imaging. The results showed that toxoplasma-positive individuals without schizophrenia exhibit particularly no reduction in gray matter density, something the toxoplasma-positive control were quite pleased about. Also in latent toxoplasmosis, high maternal levels of Toxoplasma gondii antibody during pregnancy have been associated with schizophrenia in the offsprings, while there was no significant effect found in moderate levels of antibodies. Moreover, increased risk of dopamine in the brain of the infected subjects leads to autism, schizophrenia, Alzheimer's disease, and Parkinson's disease. Twain, a cat lover, an English artist, best known for his drawing of cats and kittens. In his later years, he may have suffered from schizophrenia, and his drawing of cats changed from this into this. Oh no! Obsessive-compulsive disorder, OCD, it's a common and chronic and debilitating psychotic condition that affects about 3% of the general population. This disorder is identified as unwanted and recurrent thoughts which cause marked distress. Individuals with OCD are struggling to reduce their anxiety by mental acts and repetitive behaviors. According to the World Health Organization, OCD is one of the top 10 disorders which affect people's income and quality of life. A systemic review and meta-analysis revealed that toxoplasmosis could be as an associated factor for OCD 2 times. OCD, with its related symptoms, is more pronounced in Toxoplasma-infected individuals. Let's shift gears and move to Toxoplasma gondii-induced bacterial dysbiosis in the gut during acute phase of infection. Toxoplasma gondii invasion of the epithelium in the small intestine leads to intestinal inflammation, dysbiosis, tissue necrosis, followed by spread of the gut bacteria to peripheral organs and subsequent sepsis. Altered taxonomic composition of the gut microbiota during the acute Toxoplasma gondii infection is marked by elevated abundance of gram-positive bacteroidetes at the expense of decreased firmicutes. The inflammation resolves from the ileum. The infection progresses from acute to chronic stage. Firmicutes become the most abundant phylum, accompanied by a decrease in the abundance of proteobacteria, the gram-negative taxa, that lead to substantial dysbiosis and intestinal pathology. The effect of both acute and chronic Toxoplasma infection on the gut microbiome can be varying outcome. The results are highly dependent on the following: the genetic background of the host and the parasite, the host age and gender, the route of infection, the anatomical location of where samples were collected from within the intestine. Changes in the commensal population, notably an outgrowth of Clostridia species, were sustained in Toxoplasma chronic infection. Several studies also reported chronic enrichment in taxa, Verrucomicrobia, Akkermansiaceae in animals with chronic Toxoplasma infection also. The behavioral changes induced by Toxoplasma gondii has been a focal point of research with rodents and humans, showing altered behavior that both host at risk and provide survival advantages for the parasite. Now, we need to connect the dots between different mechanistic approaches of Toxoplasma gondii infection and bacterial dysbiosis. The direct infection of the CNS and the parasite tropism to immune-privileged organ are notable considerations. The underlying mechanism driving these changes remain unclear. Neuroinflammation, changes in the neurotransmission, and endocrine signaling stand at the piece of the puzzle, awaiting precise connection that the gut microbiome and the gut-brain axis may be able to clarify. In fact, the gut-brain axis serves as the vital conduit for communication between microbiome, gut, and brain, orchestrating an array of interactions through immune, neural, and endocrine routes, which we will discuss very soon. There is a well-established relationship between the immune system and the gut microbiota. The immune response to toxoplasmosis involves a complex interplay of cytokines, chemokines, and lymphoid cells, activating the host defense mechanisms, which evolve in a parasite-induced dysbiosis in the gut bacteria, in particular proteobacteria outgrowth, to combat Toxoplasma gondii infection at the early stage. Due to an intricate and continuous communication between the gut microbiota and the immune system, the outcome of this conversation doesn't leave the neurosystem unaffected, and chronic changes in the communication can manifest in behavioral changes. Now, what's the relation between bacterial dysbiosis and neurotransmission? In fact, gut-brain axis profoundly influence the central process such as neurotransmission and behavior. Gut bacteria contribute to this dialogue by releasing an array of neuroactive compounds such as dopamine, glutamate, and gamma-aminobutyric acid GABA. These neurotransmitters seem to play an important role in the microbial ecology, adding complexity when it comes to understanding the exact mechanisms by which intestinal microbes communicate with the CNS during Toxoplasma gondii infection. Dopamine, Dopamine is related to motivation, reward, and euphoria. But alteration in the dopaminergic transmission has been related to severe CNS disorders like anxiety. Alteration in the gut microbiota impact dopamine signaling in the hippocampus and amygdala. The dorsal hippocampus is the center of learning, memory, and spatial navigation in the brain, while the ventral hippocampus is associated with the emotional and motivational consequence of stress, including depression and anxiety. Hippocampus communicates with the subcortical structure like amygdala and the striatum, located deep within the brain's temporal lobe and is responsible for regulating our emotions, especially fear and anxiety. It acts as an emotional switchboard, receiving information from our senses and determining whether a stimulus is a threat or a reward. This remarkable structure plays a crucial role in our survival and our overall emotional well-being. The amygdala is also involved in emotional behavior, participates in fear modulation, fear-associated memory, and attention behavior that are associated with Toxoplasma gondii infection. Several bacteria, Escherichia coli, has been reported to produce dopamine in the gut. It's also known that acute Toxoplasma gondii infection leads to proteobacteria outgrowth, which is an oral infection is likely to be E. coli. Excess of E. coli could contribute to the production of excess of dopamine in the gut. Excess dopamine can go through auto-oxidation that leads to generation of reactive oxygen or nitrogen species, which in turn might contribute to necrotic pathology of the small intestine during acute toxoplasmosis. Excessive dopamine can also signal the CNS through the gut-brain axis. On the other hand, Hatter et al. Reported Clostridia outgrowth during chronic Toxoplasma gondii infection. Notably, metabolites produced by the pathogenic Clostridia species can inhibit the conversion of dopamine to norepinephrine, resulting in elevated levels of dopamine. Other evidence for a complementary role for gut microbiome in the Toxoplasma gondii infection come from studies of probiotics such as Lactobacillus and Bifidobacterium. These probiotic cocktails were found to reduce anxiety-like behavior in rats and mice. Groups have also detected Akkermansia enrichment in the gut of chronically infected mice. Akkermansia has been considered the next generation probiotic, one that may have significant impact on the brain. If chronic Toxoplasma infection leads to an outgrowth of bacteria with probiotic potential in the gut, that would probably affect the brain. In the GIT tract, dietary glutamate not only serves as a major source for glutamate, but also the most abundant 8 to 10% among dietary amino acids. Interestingly, chronic toxoplasmosis has also linked to lactobacilli overgrowth in mice. Several lactobacillus strains have the ability to produce glutamate. The brain is not exposed to excess glutamate due to the low concentration of glutamate reaching the systemic circulation, while dietary glutamate does not cross the blood-brain barrier under normal conditions. Under altered gut microbiota can cause changes in the barrier permeability, as we saw in toxoplasmosis, which can compromise the barrier and lead to transfer of the luminal glutamate into the CNS. Homeostatic control of extracellular glutamate prevents its accumulation, and excess extracellular glutamate results in excitatory, including excessive postsynaptic excitation, which has been linked to chronic inflammation. This suggests that there is an increased risk for glutamate-mediated excitotoxicity during Toxoplasma gondii infection, which can potentially contribute to cognitive impairment. Glutamate has also shown to increase the pathological conditions, such as irritable bowel syndrome (IBS) and inflammatory bowel disease (IBD). This increase in the glutamate permeability has been shown to result in altered neural responses that are not local to the enteric nervous system, but also remotely in the CNS by utilizing the microbiota gut-brain axis. Elevated psychiatric comorbidity of the development of IBS and IBD may be explained by the impaired glutamate metabolism across the microbiota gut-brain axis. Notably, both of these functional GIT disorders has been modeled by Toxoplasma gondii infection. GABA is the main inhibitory neurotransmission in the CNS, and its balance has been associated with a number of disorders including anxiety. Some gut commensal bacteria produce GABA, including Bacteroides, Bifidobacteria, and Lactobacillus species. Toxoplasma gondii infection appear to alter the inhibitory function of GABAergic signaling in mice through changing in the distribution of the enzyme GAD67 that catalyze GABA synthesis in the brain. GABA hypofunction could decrease GABAergic activity and consequently reduce neural inhibitory control and contribute to excitotoxicity. Yet, GABA-producing Bifidobacteria adolescens strain reduce serum glutamate levels in mice. Several experiments indicated that the gut microbiome affects the level of GABA and subsequently influence mental health. The 10th cranial nerve, the vagus nerve, with its afferent fibers, delicately detect the metabolites, including neurotransmitters produced by the microbiome in the gut, and conveys this critical information toward the CNS. In fact, exploring the role of the vagus nerves during Toxoplasma gondii infection holds potential opportunity to understand how Toxoplasma gondii utilize this neural metabolization of the gut-brain axis to influence behavior. Let's move to the impact of bacterial dysbiosis on hormone system. Actually, the neuroendocrine system and microbiome interact to influence social behavior. Males and females exhibit extended pattern of energy and nutritional requirement across the lifespan. Differences in the sex hormone can contribute to differences in microbial diversity and gut microbial composition. The gut microbiome plays a crucial role in testosterone metabolism in mice. The microbiome is composed of many microbial taxa, bacteria, fungi, viruses, archaea, protozoa. Yet, most studies still largely focus on the measure of bacteria. Surprisingly, the fungi, which are the taxonomic powerhouse of potent instigant of dysbiosis, are often overlooked. During times of good health, the body fungal resides, known as the microbiome, exists in harmony with other microbiota taxes. Many fungal species, however. A rule for opportunistic fungal species such as Candida albicans as drives to Toxoplasma control altered behavior and brain biochemistry has not been extensively studied. It seems logical that not only would an acute infection of Toxoplasma gondii infection introduce a pro-inflammatory intestinal environment framed for bacterial dysbiosis and dominance of fungal pathogens, but that this dysbiotic state are related functional deficit that would maintain chronically until dysbiosis was resolved. In studies of fungal translocation in human neuropsychiatric disorders, level of antibodies to fungal species like Saccharomyces cerevisiae and Candida albicans were constantly elevated versus those from comparison group. In conclusion, cats that loved by children and adults are capable of transmitting a disease that does not threaten your life by death, but increase car accident and change your personality profile. Mass personality modification regarding culture aspects that relate to ego, work, rules, money, material position affects national culture. Conclusion, gut microbiome studies are currently flourishing, although we are still very much in the beginning stage of fully appreciating the complexities of this developing field. Studies to date have to understand the potential role of fungi, which can quickly displace bacteria, create pro-inflammatory GIT environment, and generate deleterious consequences on the brain function and behavior. Acute and chronic infection of Toxoplasma gondii very likely accelerate the deterioration of the microbial balance, impacting a partnership with fungi that is mutually beneficial. Empirical studies to test co-occurrence and functional co-association of these exposure could help determine the extent that these infections are related. Now I'm going to leave you with the take-home message: prevention and control. Don't allow your cat to go outside your home. While you are pregnant, allow another family member to take care of your cat. Cover up children's sandbox. Do not allow your cat to eat raw or insufficiently cooked meat. More tips for yourself: avoid eating of raw or insufficiently cooked meat. Wash fruits and vegetables carefully. Wash your hands thoroughly. Avoid eating raw eggs or drinking unpasteurized milk. Do not rub your eyes or face while preparing food. Then wipe the counter with clean after that. Avoid flies and cockroaches in your kitchen. See, all the problems caused by toxoplasmosis could be prevented and could be easily diagnosed with just a blood test for yourself and for your cat. So enjoy your life, enjoy your cat. Thanks for your attention. Greetings from Egypt. Have a successful conference and a great day. Bye. هانم خاطر.